Welcome back to ZOO3649, lecture 15. We're continuing with inheritance and we're going to talk about Mendel's laws. A lot of this is refreshing from 2544 last year, so you should watch the video of the lecture just to brush up your memory from last year, okay? But be very sure that I'm not going to be asking you easy questions like you got in 2544 about these and about the next lecture, the deviations from Mendelism. They will be examined, but you're not going to get easy questions. You're going to have to think about your questions, okay? And there are a lot of problems on Mendelian genetics in your practical guide. Please start filling them out. And if they're not filled out, you're going to suffer for your practical mark, okay? I'm telling you now. Fill these lectures, while you might think it's so easy, it's second year. They belong to practicals 2, 3, and 4, and 5. So you better start doing those problems, okay? Because they have to be done by the time you are coming for face-to-face lecture, practical 6. So please, this is not a holiday. All right. So so dominance and recessiveness, right? So this is obvious, right? So Darwin said that there was a gene that was dominant over a dominant allele, which was an allele which was dominant, and an allele which was recessive, okay? And if you look at the purple flower gene, the P gene, okay, all organisms have to have 2 alleles, one from each parent, right? So a purple flower would be big P, and a white flower would be small p. Both those genotypes are what? Homozygous. And they're homozygous because what? Both have exactly the same allele, right? So big P, it's both big P, right? And small p is both small p, so they are homozygous. Big P will be what? Heterozygous, okay? So, in the experiment, he mated a big P flower with a small p flower, and the offspring in the F1 generation were all purple. Obviously, you are saying, I write here, I ask here how at the end of the slide, but you already know how because you're in 3rd year, okay? It happened because of this, right? So it happened because if you separate the gametes into small p and big P, and then you get them together, every square has to have a big P and every square has to have a small p. So all the next F1 generation are going to be big P, small p. They're going to be heterozygotes, but they have a purple color. That's saying that the big P is dominant over the small p. So if you have a big P in your genotype, you will automatically be purple. Even if you have a small p. Even if you're a heterozygote, you have a small p there, you will still be purple. You will not be halfway in between. You will not be a blend. Okay? So you will be purple. So Mendel thought, why is that? I mated a white flower with a purple flower. Why do I get all purple offspring? So he had the very clever idea to mate the F1 with each other. In other words, these big P, small p's, mate them with each other and see what happens. Okay? So he did that, and what did he get? As you know from 2544, he got 3 purple and 1 white. Suddenly, he recovered the white phenotype when he mated the F1 generation with each other. Okay? So the ratio was 3 to 1. Okay? He recovered the pure white small p, okay? And that demonstrating that what? Some alleles are there. In other words, you can only get the small p in F2 if F1 plants had small p, right? They can't give small p to this guy here in F2 when in F1 they themselves never had a small p. And as you know, in F1, there it is, all of them, every individual had a small p as well as a big P, right? That's why they were able to give this small p in the next generation here. Here's the small p they're giving. And in one of the squares, both of them give the small p. Okay, so the inherited allele is dominant if it can mask the phenotype of the recessive allele. Okay? If it can mask the phenotype. And that is depending on what? Again, you learned in 2544, it's depending on dosage, basically. It's depending on dosage, gene dosage. So if you have a gene which is dominant, it creates, that allele creates so much gene product that it's enough to make up for the one, the recessive mutant, which is not making the gene product. Okay? So big P is the gene that's making the gene product, and small p is the gene that's not making the purple product. Okay? So what happens? Why is the flower still purple? Because the big P makes so much of the purple color that it masks over the non-color of the white, because white is not making any color at all. That is why it's white. Okay, so that is about dosage. It makes so much messenger RNA, it makes so many purple proteins that it just masks the phenotype of the one that doesn't make any proteins, which is the white color proteins. Okay, so it's because of the dosage. That's why you're dominant. The allele can be dominant, and that's why you have alleles which are recessive. Okay, and let us talk about then segregation. We've looked at dominance and recessiveness, and I explained it to you in the last lecture. Please go back to the last lecture. But basically, segregation is explaining how you can go from each parent. Mom has 2 copies, alleles. Dad has 2 alleles from their parents, right? How do these alleles, which ones are given to you? Because they can't give all to you. You don't have 4, right? You also have 2. So what happens inside mom and dad? During meiosis, they have to segregate into 2 gametes. And only one is going to go to you from dad's side. Mom's side, same, right? Segregate into 2 gametes, and only one of these gametes is going to go to you. So of course you get 1 from mom and you get another 1 from dad. That's why you also got 2. But you could not get 1 and 1 from dad and mom if they didn't segregate inside mom and dad first. Okay? So segregation happens in the parent during meiosis. So each offspring receives only 1 copy. I've explained all this to you. This is just basically the same as what I've just explained to you, right? So that is segregation. Independent assortment is also quite it's a concept you might have had difficulty with in second year, but in third year, you need to know what independent assortment is. It's not rocket science, okay? If you can't understand this, please go and do something else. Don't do biology. This is really, really quite simple, and you need to get your heads around this, right? So, independent assortment is talking about chromosomes, right? You know that they're chromosomes. You know that they line up at the equator during meiosis, and then what? They go to either pole. They split, and one goes to one pole, and the other goes to another pole, right? That is when they segregate. But it depends how they line up. Okay. Now let us take the first possibility. Now we're only looking at two pairs of chromosomes. Okay, 1 pair here, 1 pair here, and remember how many pairs of chromosomes are there in the human body? There are 23, right? There are 23 of these pairs, but we're not showing all 23 here. We're just showing 2 so that you can understand a simple drawing. Okay, if you put 23, the drawing becomes too complicated, right? So let's just assume that there are 2 chromosomes here. Okay, and how do they line? And they have to line up at the equator, right? And then what happens? They both get split into 2 gametes. All right, and then that's metaphase 1, metaphase 2 meiosis, and the separation into individual gametes here. All right? So now, look at how these chromosomes have aligned up. Of course, this one must align with that one because they are the same chromosome. This is maybe chromosome 1. This is chromosome 2. They have to line up with each other, right? But look at where they line up. In this section, the equator goes through both chromosomes, right? Right? So, actually, the equator will go through here, right? And these two will be in this cell, and these two will be in this cell. So, in other words, in this situation, possibility 1, the one that, say mom gave you red and dad gave you blue. So the two red ones that mom gave will be in the same cell here, daughter cell here, and the two that blue that dad gave you will be in the daughter cell here. But that's not the only way they can line up on the equator. What if they line up like this? What if they line up where, okay, mom's on the left, dad's on the left, mom's on the right, but on the second chromosome, it's no longer the dad's one, which is under dad's one. Here you got mom's chromosome under dad's chromosome here. Okay? So they've lined up opposite to how they've lined up in possibility 1. And here what will happen? The equator will form in the same place, right? But you see the daughter cell will be now one from one parent and one from the other. Yeah? So depending on how they line up here will tell you what combinations of mom and dad chromosomes you get in these daughter cells. And then eventually what combinations you get in the gametes, right? But these can line up the same, but they can also line up differently. Okay, and now you can understand if you have now a third set of chromosomes here, a third chromosome, that can line up differently again, and a fourth, and a fifth, and a sixth. And so basically what Mendel was trying to say with independent assortment is that if there's a trait for round color, a round shape on this chromosome, and the shape for, and the gene for a yellow or green color on this chromosome, those two traits they are inherited independently of one another. Okay? And he was lucky enough to choose traits that are actually on different chromosomes in the P. Otherwise, he would never have figured this out. Okay? So therefore, the offspring will get a random selection of maternal and paternal chromosomes. It doesn't always line up like this. It can line up also like this. And when there are 23, these are only showing 2 chromosomes. When there are 23 chromosomes, all of them lining up together, there can be how many combinations? That is why independent assortment, we said, also leads to variation, right? Because of the number of different combinations. All right. So Mendel only discovered this, yes, when he was looking at more than one trait, and he was lucky that the both traits were on different chromosomes. Because if they were on the same chromosome, he might not have figured it out. Okay. So in order to observe the law of independent assortment, he had to perform a dihybrid cross using 2 traits. Okay. So he used the yellow color, big Y, which is dominant, and small y, you get, if you get small y, you are green. If you have big Y, any big Y, small y, or big Y, you are yellow because it's dominant. So shape, round is dominant. So if you have one big R, even if you have a small, big R, you are round. If you have a small r, you are what? You are wrinkled. Here's wrinkled, okay, and green. Here is round and yellow. So round is dominant, yellow is dominant. So this one is got big R, big Y, this P here. This P, you see wrinkled is recessive and green is recessive. So that means he's got small r here for the wrinkled and small y for the green because he's not yellow, right? So if we mate these two, what will the gametes, well, if we mate these two, what will the F1 generation look like? Yeah, they will be all round and all yellow. Why? Why? Because here are the two gametes. what gametes can segregate here? How many different possibilities of gametes can you get here? You have to have an R and a Y together, right? So what R's can you put a small r and a small y together here? No. You can only put a big R and a big Y. And this big R with a big Y, which is the same thing, right? So the only gamete you can give if you are this parent is a big R and a big Y. That's all you can give to your offspring, because that's all you have. How about this parent here? What can this parent give to the offspring? It can only give a small r because it has no other kind of r, and it can only give a small y because it has no other kind of y, right? So the small parent can only give a small r and a small y. So if you do a Punnett square with these two genotypes, what do you get? You get big R, big Y, small. So basically, for R, you are heterozygote, and for Y, you are heterozygote. In other words, dominant, round, dominant, yellow. Okay? So those are the two gametes. This is the heterozygote here. The offspring. All the offspring from F1 are round, yellow, having this genotype here. Big R, small r, big Y, small y. All of them do. It's the same like in the purple flowers, purple and white. If you mix the F1 generation, the big P, small p, what do you get? You get 3-to-1, right? You get big P, you get big P, small p twice, and you get small p, right? It's exactly the same here, right? So when you mate these guys, which are the heterozygotes, with each other, what do you get? Well, we have to do a dihybrid cross. And this is a little bit more complicated than a monohybrid cross, but let's do it. First, when we do our dihybrid cross like we did with this one, we got to figure out what alleles can these guys give. So, what combinations of alleles can be given by all these individuals which are in the F1. So you can have a big R, big Y, the same as this one. You can have a small r, small y, the same as this one. So these individuals can give the very same two alleles as their parents. But what else can they give? They also can combine a big R with a small y, like this one here. Big R, small y. They can also combine a small r with a big Y. And here it is, small r, big Y. So any parent that has this genotype, this heterozygote genotype here, the genotype of the F1, the heterozygote for both traits, any individual can give any of these four alleles. Okay? So what do we do with these alleles? We graph them on one axis, and on the other we make a Punnett square with these four alleles. And what does that look like? Let's do a dihybrid cross. Here are the alleles. Okay? And then here are the alleles again, the same alleles. And so it's a big Punnett square, right? It's not a small Punnett square, it's a big Punnett square. So here are the alleles here are the alleles here. Let's combine them. And you get big R, big Y in this corner. And in this corner you get small r plus small r and small y plus small y. So you got small r, small y, small y. And you get different kinds of genotypes depending on how which gametes are coming together. Okay, so you figured out what the gametes are going to be. You've then worked out the combinations. And what's the proportion? The proportion of each phenotype. You have how many phenotypes here? You've got, and you can see the phenotypes with your eyes. You've got round and yellow. So basically this was the phenotype of the F1 generation. All of them were like this. But in the F2 generation, you don't get all of them like that. Just the same as you did with the monohybrid cross. You recover the recessive phenotype. Okay? And in this case, we are also recovering. Remember, all the parents looked like this. We mated these guys with each other. All of them looked like this. But now suddenly, some of them look like this. Some of them look like this. And some of them look like also this. Okay? It's a dihybrid cross, so you have the combinations as well. So what are the proportions? 9, 3, 3, 1. 9 is what? So we've got round yellow. How many round yellows do we have? 1, 2, 3, 4, 5, 6, 7, 8, 9. So that's the 9. How many round greens do we have? 1, 2, 3 round greens. That's the 3. How many round wrinkled yellows do we have? 1, 2, 3. That's the other 3. And how many wrinkled greens do we have? Just the 1, and that's the 1 there. Okay, so you see how you get that 9 to 3 to 3 to 1 year ratio. This is all second year stuff, guys. You know all this already. How about probabilities? Now, this is directly leading to your practicals, right? You need to understand these things to do your practicals properly. So I'm asking you, how do you work out probabilities? If I asked you what probabilities were of different genotypes, what would you say? And there's a lot of questions like this in the practical guide. Okay, please, you got to go through them. So probability of 0 means something will never happen. Like, what is the probability of 0? The probability that it will snow in Venda. Okay, that's a probability of 0. It's not going to happen, right? Whereas the probability that it will rain in Venda, yeah, that's a higher probability. It's not 1. If it was 1, it would be always raining in Venda, right? But the probability is higher than 0 that it will rain in Venda. Okay, so, and the probability of 1 means it always happens. So, what is the probability of a round and yellow phenotype? Okay, where's round yellow? Round yellow looks like this. How many look round yellow? 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, there are 9 round yellow. You divide that by the total number of squares, the total number of possibilities from your Punnett square, which is 16, and that is the probability. Okay, that is the probability of your round yellow phenotype. So if I had to ask you the probability of the wrinkled yellow phenotype, what would you say? You would say, you would count wrinkled and yellow. Where are they? 1, 2, 3. And it would be 3 divided by 16. So the probability of a wrinkled yellow phenotype is 3 over 16. The probability of a wrinkled green, and there's only 1, it's 1 over 16. Okay? Really easy. Now it comes to test crosses, and you know already what a test cross is. A test cross is when you don't know the genotype of something, but you want to find out. Okay? So in that situation, you always, so if you don't know the genotype of a particular individual, you mate it with the individual that you do know. Okay? And then you mate them, you see what the offspring phenotype is, and from the offspring phenotype, you can figure out what was the genotype of the parent that you didn't know. Okay? And that is what you call a test cross. Okay? In this case, right, you don't know the genotypes of the round yellow dominant individuals. Okay? Say, for example, you didn't know the phenotype of this guy here. You could see, but you didn't know. Because, for example, that's the genotype. You didn't know the genotype. Because let's face it, right? If you are round and yellow, we've counted them. There are 9 of them here, round yellow. But they don't have the same genotypes, do they? You can have big R, big Y, and be round and yellow. You can have big R, big Y, small y, and still be round and yellow. And you can have big R, big Y, and still be round and yellow. Right? So you can be different genotypes, but still have the same round yellow phenotype. So now, if I have a round yellow pea, and I say to you, what's the genotype of this? You won't be able to tell me, right? Because you won't know which of these different genotypes you see there can be the one for your round yellow, because round yellow is coming from different genotypes, as you see there. Now, so what is the genotype of a round yellow? How do you figure that out? What do you mate him with? You have to mate it with something. How do you figure it out? You would cross each individual that you don't know, right, with a known homozygous recessive, and then look at the offspring, what phenotype the offspring has. Okay? So a test cross always has a homozygous recessive that you cross it with. Okay? Because that's the only one that will tell you the answer. Okay? So you don't know what you don't know a particular individual's genotype. You mate it with what? A homozygous recessive, always. And then you can figure out what that guy's genotype is by looking at the offspring. Okay? Because then it'll be obvious what the offspring is. And why do we always use a homozygote? Because if we didn't use that, we wouldn't be able to tell. So for example, if you mate this homozygous recessive with this round yellow, okay, I can see immediately that not one, I can see if you mate this one with this one, look at the alleles. I can tell you now that not one of the offspring is going to be anything but round yellow. Okay? In other words, if you made this one, say it was this one you didn't know, and then you used this homozygous recessive to mate with it. The offspring between this one and this one, not one will be green, not one will be wrinkled. All of them will be round and yellow. Why? Because all of them will be big R, big Y, small y. Right? All of them will look like this. Big R, big Y, small y. Okay? If you made these two together, you will only get this, right? So then you know, oh, all my offspring are round and yellow. That means what? That means what? That means the unknown one was a homozygous dominant for both round and yellow. However, what if the round and yellow was this genotype? No? If it was this genotype, but you don't know the genotype, you want to find out the genotype. So you do a test cross with this guy and who? Again, always the homozygous recessive. What will you get here when you put these alleles? I can tell you already you will get a green color because this Y here, small y, and this small y will come together. You will get at least one green. And I can tell you also you will get at least one wrinkled because this one has a small r and this one has a small r, and those two small r's will also come together. So you will also get a wrinkled. So now immediately you made this one round yellow. You don't know what is its genotype. You made this one with the known genotype homozygous recessive, and what do you get? You get individuals that are not only round and yellow. You get some wrinkled and you get some green, and then you know, this one here, it was this genotype here, because if it was that genotype, I would not have gotten any green. I would not have gotten any yellow. Okay, so that's why you use this homozygous recessive to tell the difference. Okay, so in summary, I don't really have to go through that. You can read it for yourselves. And we've been discussing this now for a long time. You got this also in second year, so I think there's no real need to repeat and repeat and repeat. I think you've got it now. You should have it now. Okay, so the next lecture will be on the last lecture in this section, and we will look at situations where you have inheritance, but you don't have Mendelian inheritance. So the inheritance deviates from a strict Mendelian pattern. And a Mendelian pattern is what? When it's one trait, it's 3 to 1, right? When you have two traits independently assorting, you have 9, 3, 3, 1. Okay, that's the Mendelian pattern. But there are some genes that don't have the Mendelian pattern, and that's what we're going to deal with in the next lecture. Okay, I'll see you then.